OS. If we were to look at a triglyceride, this is our basic fat. Are we good on saturated or unsaturated or not? Do you want clarification? Okay, so let's say this is a fatty acid tail, and this fat has 3 of them. Let's say all their tails are kinky. All right? Kinky tails, are those unsaturated or saturated? Unsaturated, because they have all these double bonds in it. What could I add to it to saturate them? Hydrogen. And if I add hydrogen, then hydrogen goes around the whole thing, and now we have saturated fats. And as a result, they stack, and they're going to be a solid at room temperature. Oils are going to be unsaturated. Solid fats are going to be saturated. And we can transform them. We can get a trans fat where we're actually switching between the two. Here's phospholipids. This would be spontaneously how they form, but this is in a cell membrane. All right? Water here, but no water in the middle. Is water polar? Yes. Are the tails of phospholipids polar? No, but the heads are, and so they're going to be attractive. And then do we remember what cholesterol does? Are we good on that? Yeah, so it mediates during temperature, right? When they get hot, they spread apart. When they get cold, they get together. And so cholesterol is going to fit in the middle. Yeah, why is it sitting there by the fatty acid tails, but not by the heads? It's nonpolar, right? It's a lipid. Cholesterol, phospholipids, triglycerides. And so the only part of all of these that's going to be polar is going to be the heads. All right. Ready to go to proteins? Yes. Okay, building blocks? Amino acids. Where do we chop them down, Joel? Where do we chop down the proteins into their little amino acids?
Oh, in the
digestive system, yeah. Where do we build them up in a cell? Because Tuck said it's in the cell. Where specifically do we build them up in the cell? Ribosomes, okay? So you take in the amino acids, you can make them into anything that your heart desires. So let's look at these. So these are going to be 5 different amino acids. Don't be scared, because there's chemistry. But can you see the things that are the same? The carbon in the middle. We've got an amino group on the left side, and we have a carboxyl group. So now it looks less scary. They're all the same, except for, do you remember what it's called that goes off the bottom? R group, yeah. So the R group is going to be different in each one of these. Don't memorize any of these, because I don't know them either. I have to look them up. All right, but let's look how they're hooked together during dehydration synthesis. Are you good on what that means, dehydration synthesis? So we're pulling out of water. And so let's position one over here, and then we move this one next to it. And can you see the water that's right there? So that's going to go away, and what's going to come in its place? A covalent bond between the two. And now let's put another one there. Can you see the water? And now we have a covalent bond. This is taking place, we said, in the ribosome. And another one goes there, we lose the water. And another one goes there, and we lose the water. And now we have a bunch of amino acids attached together. We call that a polypeptide. Yeah. Let's not call it a protein yet until it folds. Yeah, that's the big thing. So where is this occurring? In the ribosome. If all this stuff looks scary, we'll spend a whole level just on this, on how the ribosome comes together and how the mRNA goes through it. But here's going to be all those amino acids that are streaming off the top. Now, some of those R groups are going to be polar. Some of them are going to be non-polar. Some of them are going to be negative, and some of them are going to be positive. And as a result, when we fold a protein, we're going to get all these different interactions between the R groups. So let's say it's a hydrophobic. Where's that going to be? On the inside. And hydrophilic? On the outside. And if we have a positive here and a negative here, they'll attract. And so that's where we get all these different connections. Yeah, Mary?
Another thing sort of on what we're supposed to know.
Say it again.
Another thing sort of on what we're supposed to know. Is there any way to like easily tell what it is?
If polar or not. Yeah, I know a little bit about chemistry. So for me, I would say this one, if you ever see an oxygen like this, it's going to be greedy, and so it's going to be polar. If we don't have any oxygen or nitrogen, generally, this is almost like a little fat, so it's going to be non-polar. Yeah. But you're not going to be expected to do that, for sure. Yeah. Do you remember I played that game, Fold It, for a second, and we had to pull these apart, so the R's to make them happy? And so basically, what do we get as a result of this? This is why I turned the lights off. We get primary, secondary, tertiary, and then quaternary structures. And that's kind of a dark slide. So what's primary structure? Amino acid sequence, right? The order the amino acids come in. What's secondary structure? Alpha helices and beta pleated sheets. Anybody, help me out. Nobody's got this right today. What gives the alpha helices and their beta pleated sheets their structure? Yeah, do you know? Yeah, so these little dots are going to be hydrogen bonds that hold it in place. What's going to give us the tertiary structure? Yes, yell it out. I can't even see you. R group. So it's going to be all the R groups that come off the side. And then are we good with quaternary? Are multiple proteins coming together, so subunits coming together? Yeah. Now, this is a polypeptide, and that is a full-fledged protein. Okay. Let's go to nucleic acids then. This is the one that we know least about. Yeah?
So when you have a polypeptide, that's what the ribosome makes, right?
Uh-huh.
And then is that what, does it turn in?
Yeah, we saw it in the video. I don't know if you caught that yesterday, but there was like a ribosome, and RNA was going through it, and as the polypeptide was coming out, it was folding right as it comes out.
Okay, and then what happens when it, you don't have to know, but what happens when it goes into the Golgi apparatus? Does it switch more? Or
Yeah, we're going to add stuff to it. And so we're going to add things, yeah, mostly carbohydrates. We're going to add sugars and tags on the outside. Sometimes lipids, but yeah. Good question. So let's go to nucleic acids. We're good on RNA, DNA. Could we delineate between the two? I mean, just looking at them, DNA is a double helix, right? RNA is a single helix. Can you go deeper? Anybody know differences between the two? Yeah,
The RNA has a
uracil instead of a T. Yeah, anybody, there's one more? The sugars. And so this one's going to have a ribose sugar, and this is a deoxyribose sugar. We'll come back and visit DNA and RNA again. Just a quick side note, DNA sits inside, let me go back. DNA sits inside the nucleus, and it's really lame. It just has the directions and the genes. RNA is the worker. RNA does everything else. It takes the message out, transports it. We're finding right now that RNA, the more we look at it, is way more important than DNA. Especially little things called microRNA. It's just crazy what it does. But let's go through the parts of a nucleotide, because almost everybody misses this in the review. So there are 3 parts in a nucleotide. This right here is called the phosphate. That's the sugar, and then we're going to have a nitrogenous base that goes off the side. So that's something we should know, basic structure of a nucleotide. If you don't know that, number one, that those are what nucleic acids are built of, and that this is their structure, make sure you get that quickly learned. In everything I've talked about up here, we've got basically everything's built of carbon. So even on this model, all the gray you're looking at is carbon. They don't even show you the hydrogen because it's everywhere. But if we were to look at the backbone, this is a phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. The backbone is all the same. But if you look in the middle, can you see all the blue going down the middle? That's going to be the bases, the nitrogenous bases. And there's going to be a lot of nitrogen on the inside. And so you can see the nitrogen right there. So let's look at the 4 basic nucleotides in DNA. Can you see the differences? What are the major differences? Size of the nitrogenous bases. Did you see that? This is like a double ring, and this is going to be a single ring. So these are what we could call purines, and the other one's pyrimidines. But if we look at how they connect together, so what's the structure and how they bond are the big important things. So the first one, they'll line up, we lose a water here, and we form a bond. Does that make sense? Dehydration reaction? Let's put another one here. We lose a water, we form a bond. We lose a water, we form a bond. And so they're going to stack right up on top of each other. Can you visualize how that's like RNA now? It's just a single strand. What's the only difference? In RNA, we have a uracil here instead of a thymine. But do you know how the DNA bonds? It's a little different. So you have them on top of each other, but what we get is that. Oh, that's weird. So they'll go upside down, and then they'll form hydrogen bonds between the two. And this one will go like this, upside down, and then we get a hydrogen bond between the two, and then we get a covalent bond right there. So that's how DNA is going to bond. We're going to bond vertically, and we're also going to bond horizontally. And that gives us intense structure in the middle. Questions on nucleotides? Question on nucleic acids? We'll come back and visit this again, I promise. DNA is super important. We'll come back. All right, let's keep going then. DNA, RNA, let's go. So if we look at this, how could you do on all those labels up there? Could you label them all? Maybe? Okay. What's the inside? Cytoplasm. Say it with me. What are we going to call these spindly things in here? Cytoskeleton. What's the blue way up here? Outside the cell? Do you know what these things are? Extracellular matrix. So that's going to connect cells to cells. Let's go easy then. What are these things? Phospholipids. What's that? What's that? What's that? What's that? Oh, you guys are good. Yeah. Okay. What do these do? Form the membrane. What do these do? Threefold. Transport, receptor, enzyme. Those are the big 3 things that they do. So proteins. And in the review, that was a big one that was missed a lot. So what do the proteins do? They transport material. They serve as a receptor. They can get signals outside of the cell. And then the final thing they can do is they can act as an enzyme, so they can break material down. Then we've got